DNA Depot welcomes you to What is in Our Water, Kit DD303. In this experiment, you will learn how to test water for coliform and E. coli bacteria using a reagent that will change color when these bacteria are present. You will learn that positive and negative controls are necessary when performing experiments so that your results are accurate and reliable. This experiment is not to be used to determine the potability or drinkable quality of water. Please read all directions before performing any experiment. This kit contains the following items. 1. 15 milliliter capped tubes to be used for collection of water samples from the environment prior to starting the experiment. 2. Smaller tubes that hold beads which contain E. coli and coliform bacteria. These will be used as positive control water samples. 3. 4 2 milliliter micro test tubes that will be used to test the water samples you collected and the negative control water sample. 4. A bottle of coliform detection broth. 5. A bottle of sterile water which will be used as the negative control water sample. 6. Sterile transfer pipettes that will be used to place water samples and coliform detection broth in the test tubes. In addition, you will need the following items. 1. Disposable gloves. 2. Water samples from the environment, such as from a fish tank, local stream, or well. 3. Disinfectant, such as 10% bleach to disinfect material at the end of the experiment. 4. A waterproof permanent marker to label tubes. And finally, a tube holder that can be made of rubber foam or styrofoam. In step 1, use your marker and label the 4 microtest tubes 1, 2, 3, and 4. Remove the tops and place them in or on a container that will keep them upright and secure. Step 2. In Step 2, you will be adding the reagent or coliform detection broth to the micro test tubes before adding the test samples. Carefully open a new sterile transfer pipette in a way that you will be able to place it back into the sterile packaging when done so that it can be reused in Step 4. Open the bottle of coliform detection broth and carefully add 0.75 milliliters, which is the third mark on the transfer pipette, to each of the 4 micro test tubes. Step 3. In Step 3, you will be adding a control sample that is provided and the water samples you collected to the chloroform detection broth already in the tubes. Use a new transfer pipette to add 0.75 milliliters of sterile water to the tube labeled 1. This will serve as the negative control water sample. Cap the tube after adding the control sample. The pipette can be discarded. Record tube 1 as negative control in your notebook or in the chart in your booklet provided. Use a new transfer pipette to add 0.75 milliliters of water sample 1 to the tube labeled 2. Cap the tube after adding the water sample. Discard the pipette. Record the source of the water sample in your lab notebook or in the chart in the booklet. Repeat this step for the other 2 water samples you collected. Remember to record the source of the water samples for tubes 3 and 4. Step 4. In Step 4, you will add the coliform detection broth to the tubes with the E. coli and coliform beads. Reuse the pipette from Step 2 and carefully add 1.5 milliliters coliform detection broth to each of the tubes with the beads. Do not contaminate the pipette, so be sure you do not touch the pipette tip to the tubes with the beads while adding the reagent. This will serve as the positive control water samples. Cap the tubes finger tight and discard the pipettes. Place all the samples in a washable container and incubate for 48 hours at room temperature. After 48 hours, retrieve your samples. Be sure you are wearing protective gloves when handling your samples. Step 6. The expected results of this experiment should show that the negative control water sample, that is the sterile water, should remain a light yellow color. The positive control water samples that contain the E. coli and coliform beads should have a bluish-green color. Compare your water samples to the negative and positive control samples. Do any have a bluish-green color? Record your results in the lab notebook or in the chart found in the kit booklet. Did you expect to find these results? As noted in the booklet, this experiment is not to be used to determine the potability or drinkable quality of water. Upon completion of the experiment, disinfect the tubes in 10% bleach for at least 1 hour before discarding the materials in the regular trash. The disinfectant can be poured into the toilet and flushed. High levels of bacteria can cause serious human disease. This experiment simulates how to test water for human consumption. Students will use a color-based water test that yields a blue-green color for samples that harbor bacteria. Water samples are incubated with the test reagents for 24 hours at 37 degrees Celsius, or 48 hours at room temperature. Results are determined by the appearance of color. If water samples contain harmful bacteria, the test sample will turn a bluish-green color, while water samples that do not contain bacteria will remain pale yellow in color.